Very good morning, students. Now let us move on to the next topic, that is glycogenolysis. In the last class, we focused regarding glycogenesis, that is nothing but synthesis of glycogen from the glucose molecule, right? So we have seen various steps involved in that. So now let us move on to this once after the synthesis of glycogen, we have to break down, right? So the breakdown of the glycogen or the degradation of this stored glycogen, which is going to be stored in the liver and as well as muscles, right? So that process is referred as glycogenolysis. Glycogenolysis. So the breakdown of the glycogen molecule. So the degradation of this glycogen is not a reversible process, where you can make out the major form of the glycogen which is going to be stored in the liver, especially I have already explained you in the last class, and as well as in the muscles, which will be utilized during strenuous exercise, and in the liver, which is going to be stored during the well fed state condition, right? So this stored form of the glycogen in the liver, which provides glucose molecule for the various purposes, that is into the circulation it is going to be entering, is further utilized by the various tissues. Especially in the peripheral tissues, you can see this glucose 6 phosphate molecule, which is produced by the glycogenolysis, will be used up for the glycolysis process. Of course, glucose 6 phosphate is the intermediate of glycolysis. An independent set of enzymes are going to be present in the cytosol to carry out this process, that is glycogenolysis process. This glycogen is going to be degraded mainly by breaking alpha 1,4 and as well as alpha 1,6 glycosidic bond. As well, I have already taught you in the last class where will be the formation of alpha 1,4 glycosidic bonds in between two glucose residues in the linear chain of glycogen molecule, and how the branching is going to happen. I have already discussed in the last class. So now let us see what is the action of this glycogen phosphorylase enzyme. Mainly the alpha 1,4 glycosidic bonds, which are, you already know where will be the reducing end and non-reducing end. In the last class I have shown you that slide also. So the alpha 1,4 glycosidic bond, which will be present in the glucose molecule between two glucose molecule, that is from the non-reducing ends, which are going to be cleaved sequentially with the help of this enzyme, that is glycogen phosphorylase, in order to yield glucose 1 phosphate molecule. So we are going to have glucose 1 phosphate molecule by the sequential cleavage of this glycogen affected by the enzyme glycogen phosphorylase. So how this glucose 1 phosphate is going to be released? How this glycogen phosphorylase is going to act upon this non-reducing end, you can see in this slide. So this is the non-reducing end, so it is acting upon this particular non-reducing end, this glycogen phosphorylase, and you can make out those glucose molecule which are going to be, that is in the form of glucose 1 phosphate, is going to be cleaved, right? So you can see these are the glucose 1 phosphate which is going to be released from the by the breakdown of this glycogen acted upon on the glycogen by the enzyme glycogen phosphorylase. So this enzyme always acts at the non-reducing end, that is which will be present only that is going to act upon only at the non-reducing end, that is at the 1,4 glycosidic link is going to be cleaved, especially that is by this particular process is going to happen by phosphorylasis. That process is referred as phosphorylasis mechanism. So you can make out the particular inorganic phosphate is going to be present here. So you can see here, right? So that is mainly acted upon by this enzyme, is cleaved by phosphorylasis reaction or the phosphorylasis process with the retention of energy potential in the form of phosphate ester, that is will be present in the form of phosphate ester in the molecule, that is glucose 1 phosphate, in the glucose 1 phosphate, which where you can make out the phosphate will be present in the glucose 1 phosphate. So that's why this process is referred as phosphorylasis. So this phosphorylasis process is going to continue until 4 glucose residues, you can see in this slide, 4 glucose residues which are going to remain on either side of the branching point, that is at the 1,6 glycosidic bond or the glycosidic link on either side of the branching point, that is called as 1,6 glycosidic link. So the glycogen which is going to be so formed is known as limit dextrins, which cannot be further degraded by this action of this phosphorylase enzyme, that is glycogen phosphorylase. Only once after the formation of limit dextrins, the same enzyme cannot act upon that, that is phosphorylase enzyme cannot be further degraded by glycogen phosphorylase enzyme. So mainly this particular enzyme is bound with one molecule of PLP, that is called as mainly involved in the particular process which will help you in carrying out this phosphorylasis mechanism to form limit dextrins. Now once this enzyme cannot act, now the next enzyme comes to the picture, that is called as action, that is called as debranching enzyme. So what will be the action of this debranching enzyme, we can see in this slide. So what is the action of the debranching enzyme, you can see here. So you can see here the branches of the glycogen which is going to be cleaved by two enzyme activities with a which is present on the single polypeptide chain, right, that is called as the debranching enzyme. So this enzyme is bifunctional in nature, right, which will have the two functional activities, two enzyme activities on the same single polypeptide. So you can make out here, so one enzyme, that is glycosyl 4 transferase, right, that is also called as oligo alpha 1414 glucan transferase enzyme activity, which is going to remove a fragment of 3, you can make out here, fragment of 3 or 4 glucose residues which are being attached at that particular branch point, that is called as alpha 1,6 link, right. You are understanding in this slide, by making out in this slide you can make out this 3 to 4 glucose residues which are going to be attached at this branch point, we can make out, which is going to remove, right, a fragment of 3 or 4 glucose residues which are going to be attached at that branch, that is alpha 1,6 link, which is going to transfer them to the another chain by the action of the transferase activity. So the transferase enzyme is involved in the transferring of that particular 3 to 4 glucose residues to the next chain, but this glycosyl or the glucan transferase enzyme is involved in the removal of a fragment of 3 to 4 glucose residues attached at the branch point, that is called as 1,6 link. So you can see that particular transfer chain of 3 glucose to any non-reducing end. So only it can be transferred to any non-reducing end, not to the reducing end. Only at the to the site of the non-reducing end they are having capacity to transfer the 3 to 4 glucose residues by the action of the transferase activity of the particular enzyme. That's why we are going to call it as bifunctional in nature, right? Two activity is done by same enzyme. So you can see now this debranching enzyme, that is glucosidase, in that particular position alpha 1,6 link, which will help you in the removal or making free of the glucose molecule, right. So that is at the non-reducing end, you can make out here where at the branch point, right, 1,6 link. So 3 glucose residues which has been transferred, then it is acted upon by this particular enzyme glucosidase. You can make out where the particular glucose molecule you can make out, that is a free of glucose molecule by the action of this glucosidase enzyme. At the 1,6 linkage position it is getting cleaved. Now you can see once after the formation of the that is particular glycogen, sorry, what is that? Glucose molecule, free glucose you have seen, and you have seen the particular transfer of the 3 to 4 glucose residues at the non-reducing end. So now the glycogen phosphorylase enzyme, you can see here what is the action of this enzyme. So it is just help you in the removal of one glucose molecule and getting phosphorylated, that is resulting in the formation of glucose 1 phosphate, right? So this glucose 1 phosphate is getting undergoing further mutase reaction by the action of the enzyme phosphoglucomutase. So phosphoglucomutase, which is going to have an effect on this particular molecule, that is glucose 1 phosphate, which further leads to the formation of glucose 6 phosphate. Glucose 1 phosphate is getting converted to glucose 6 phosphate, which further dephosphorylated, or it is going to enter into the glycolysis. It can get dephosphorylated also, or there is nothing but removal of phosphate group, or even it can enter into the glycolysis as such, because glucose 6 phosphate is an intermediate of glycolysis. So here 1 alpha 1 4, only one alpha 1 4 bond is going to be cleaved, and the same alpha 1 4 bond is going to be replaced at different place, right? So amylo alpha 1,6 glucosidase you have seen, which is going to break the alpha 1,6 bond at that particular branch with a single glucose residues, what we have seen in the last slide, and it is going to release a free glucose molecule, right? This action of this amylo alpha 1,6 glucosidase, it is going to break the, you see here, it is going to break the alpha 1,6 bond and make and releases only free glucose molecule, right? That free glucose which is going to be released, and the remaining molecule of this glycogen is again available for the action of this enzyme, that is continuing again available for the action of the phosphorylase activity, that is glycogen phosphorylase is acting upon that, and further debranching enzyme in order to repeat the reaction. Same reaction is goes on repeating. So we have seen the combined action of this glycogen phosphorylase as well as debranching enzyme, where the glucose 1 phosphate and as well as free glucose molecule, which are going to be produced in the ratio of 8:1, and glucose 6 phosphate we have seen is getting converted, is undergoing mutase reaction to form glucose 6 phosphate by the action of this phosphoglucomutase enzyme. What will be the fate of this glucose 6 phosphate also seen depending upon the tissue, that is hepatic glucose 6 phosphate is hydrolyzes glucose 6 phosphate to glucose molecule, that's what it is getting dephosphorylated. Depending upon the tissues, the fate of the glucose 6 phosphate will be decided, that is whether it has to undergo hydrolysis, that is nothing but dephosphorylation reaction, or it can also enter into the glycolysis pathway as such glucose 6 phosphate, where here in hepatic glucose 6 phosphatase, which is going to hydrolyze glucose 6 phosphate to glucose molecule, then this free form glucose is going to be released into the bloodstream. So the muscle will not release this glucose into the blood stream because muscle tissue does not contain this enzyme action, that is glucose 6 phosphatase will not be present in the muscles as such. So where the ATP which is going to be provided for the muscle contraction via glycolysis, that's why via glycolysis we can provide the ATP for the muscle contraction. So that's why as such glucose 6 phosphate can enter into the glycolysis, that is depending upon the tissues, right? That is the fate of the glucose 6 phosphate only depends upon the tissues, whether the glucose 6 phosphate should enter into the liver or the kidney or even other intestine also contains this particular enzyme glucose 6 phosphatase except the muscles. So that's why we are going to provide the ATP via the glycolysis for the muscle contraction. We have already seen this, how this glucose 6 phosphate is getting cleaved to glucose molecule, where the presence of this enzyme in other tissues and absent in the muscle, and this will also be absent in the brain as well, that is glucose 6 phosphatase. Hence the free glucose cannot be produced. So the glucose 6 phosphate, from the glucose 6 phosphate that is entering into the glycolysis in case of other tissues can happen, right? In these tissues which we can see, from the glucose 6 phosphate it is entering into the glycolysis, and as such it is going to continue via the glycolysis to form pyruvate molecule. So that is the through the via the glycolysis that will happen. So you can see here the glycogen acted upon that particular enzyme glycogen phosphorylase, breaking of the alpha 1,6 bond link to form limit dextrins. So you can see debranching enzyme activity and as well as transferase activity, which I have explained you both. And the free glucose molecule is going to be released, and upon that you can see glucosidase activity is main glucosidase. Because of the glucosidase activity, alpha 1,6 bond is getting cleaved and free glucose is going to be released. Further, this linear form of the glycogen will be acted upon by the enzyme, the same enzyme phosphorylase, glycogen phosphorylase now, to form glucose 1 phosphate. Further, it is undergoing Mutase reaction to form glucose 6 phosphate, and further it is acted upon by glucose 6 phosphatase enzyme only in case of liver, intestine, right? Not in the brain and the muscle. This enzyme will be absent to form the glucose. Whereas glucose 6 phosphate is entering into the glycolysis as such. It mainly depends upon the tissue uptake. This completes your glycogenolysis. Please go through. This is very important. If you have any doubts, you can clear it. Thank you. In the next class, we will discuss with respect to the regulation of this glycogenolysis and glycogenesis together. Thank you.